What is SAS and what are its main components used in clinical trials? SAS, which stands for Statistical Analysis System, is a software suite widely used in the pharmaceutical industry for statistical analysis, data management, and reporting. Its main components used in clinical trials include: Base SAS provides data management, data manipulation, and basic statistical procedures. SAS Stat offers a comprehensive set of statistical procedures for data analysis. SAS Graph enables the creation of high-quality graphs and visualizations. SAS SQL allows querying and manipulating databases using SQL syntax. SAS Access provides interfaces to various external databases and file formats. SAS Clinical Data Integration. SAS Odds Output Delivery System. In addition to these core components, SAS also offers a number of specialized features for clinical research, such as Clinical Trial Management, Clinical Data Repository, Clinical Data Interchange Standards. What is Clinical SAS? What's new in Clinical SAS? Clinical SAS refers to the application of the SAS software in the field of clinical research and healthcare. SAS is widely used in the pharmaceutical industry and clinical research organizations, CROs, to manage, analyze, and report data from clinical trials and other healthcare studies. The field of clinical SAS is constantly evolving as new technologies and methods are developed. Some of the new features and capabilities of clinical SAS include SAS Clinical Trial Design and Analysis. This new product helps users to design and analyze clinical trials more efficiently and effectively. It includes features for planning trials, managing data, and conducting statistical analysis. SAS Clinical Enrollment Simulation. This new tool helps users to project enrollment scenarios for clinical trials. This can help to ensure that trials are on track to meet enrollment goals. SAS Clinical Data Integration. This new product helps users to integrate clinical data from multiple sources. This can help to improve the quality and consistency of data and make it easier to analyze data from different studies. What is the role of SAS in the pharmaceutical industry? The role of SAS in the pharmaceutical industry is crucial. It is used for clinical trial data management, analysis, and reporting. SAS helps in the efficient handling and analysis of large volumes of clinical trial data, ensuring data integrity, quality, and regulatory compliance. It allows researchers and statisticians to analyze data, identify trends, generate tables and graphs, and produce reports for regulatory submissions. Can you explain the different phases of clinical trials and how SAS is used in each phase? Clinical trials consist of several phases, each serving a specific purpose. SAS is used in different phases as follows. Phase 1, SAS is used for data collection and management, creating datasets, and performing descriptive statistics. Phase 2, SAS is employed for data cleaning, data validation, and analysis of efficacy and safety endpoints. Phase 3, SAS is used for analyzing efficacy and safety data, creating statistical tables and listings, and generating reports. Phase 4, SAS is utilized for post-marketing studies, safety surveillance, and real-world data analysis. How would you define SDTM and ADAM in the context of clinical trials? SDTM, Study Data Tabulation Model, and ADAM, Analysis Data Model, are two industry-standard data models used in clinical trials. SDTM provides a standard structure for organizing and formatting clinical trial data, ensuring consistency across studies. It defines specific variables and domains to represent data collected during the trial. ADAM focuses on the analysis datasets used for statistical analysis. It provides a standardized format for analysis-ready datasets, making it easier to perform statistical analysis across different trials or studies. What are the different types of clinical trial data you have worked with? The different types of clinical trial data I have worked with include demographic data, patient characteristics such as age, gender, and race. Clinical assessments, data collected from physical examinations, laboratory tests, and diagnostic procedures. Adverse events, information about any unfavorable medical occurrences experienced by study participants. Efficacy endpoints, outcome measures to evaluate the effectiveness of the investigational treatment. Concomitant medications, data related to medications taken by study participants during the trial. Can you explain the process of data cleaning and validation in SAS? The process of data cleaning and validation in SAS involves several steps. Identifying and handling missing values or outliers. Checking for data integrity and consistency. Validating data against predefined rules or specifications. Performing data transformations and derivations. Verifying data quality and accuracy. Conducting cross-checks and reconciliations between different data sources. How would you handle missing values in clinical trial data using SAS? To handle missing values in clinical trial data using SAS, I would consider the following approaches. Deleting observations with missing values, but only if the missingness is random or negligible. Imputing missing values using appropriate methods, such as mean imputation or multiple imputation. Creating indicator variables to flag missing values and include them in the analysis as a separate category. Employing advanced techniques like maximum likelihood estimation to handle missing data in more complex scenarios. Have you worked with data sets in SAS? If yes, can you explain the different types of data sets? Yes, I have worked extensively with data sets in SAS. SAS offers different types of data sets, including SAS data sets. These are the most common type and can store both data and metadata. SAS views. Views are logical representations of data that allow efficient data access and manipulation without physically copying the data. SAS formats. Formats define the appearance and display of data values. SAS catalogs. Catalogs are repositories that store SAS formats, macros, and other utility objects. What is the difference between a SAS data step and a SAS proc step? The main difference between a SAS data step and a SAS proc step is as follows. SAS data step. It is used for data manipulation, such as reading, modifying, and creating datasets. It involves programming statements and data transformations. SAS proc step. It is used for performing specific procedures or analysis on existing datasets. Proc steps include predefined procedures for statistical analysis, data summarization, and reporting. How do you generate tables, listings, and graphs, TLGs, using SAS in clinical trials? To generate tables, listings, and graphs, TLGs, using SAS in clinical trials, I would typically use the SAS proc report or proc tabulate procedures for tabular output. For graphical output, I would use proc sgplot or proc gplot to create a wide range of visualizations, including line plots, bar charts, and scatter plots. I would customize the output using options and statements within these procedures to meet the specific reporting requirements. Have you worked with SAS macros? Can you explain their role in clinical programming? Yes, I have experience working with SAS macros. In clinical programming, SAS macros are used to automate repetitive tasks, increase efficiency, and improve code reusability. Macros can be created to generate standard analysis datasets, perform common data transformations, or create custom reports. They help streamline the programming process and ensure consistency across multiple studies or projects. How do you ensure the quality and integrity of your SAS programs? To ensure the quality and integrity of SAS programs, I follow these best practices. Write clean and well-commented code for readability and maintainability. Perform thorough testing and validation of programs against predefined specifications or requirements. Conduct peer reviews to identify and fix any programming errors or inconsistencies. Follow standard programming conventions and adhere to good programming practices. Document the programs, including data flow, logic, and assumptions, for future reference and audit purposes. What are some common programming techniques you use in SAS for clinical data analysis? Some common programming techniques I use in SAS for clinical data analysis include data merging and joining using the merge or SQL statements. Creating derived variables using functions, formats, and data transformations. Using conditional logic with if-then statements for data selection or recoding. Aggregating data using proc means or proc SQL for summary statistics. Applying statistical procedures like proc GLM, proc logistic, or proc mixed for analysis. Have you used SAS for regulatory submissions? If yes, can you explain the process? Yes, I have used SAS for regulatory submissions. The process typically involves creating analysis datasets following CDISC standards, performing statistical analysis, generating tables, listings, and graphs, and documenting the entire process. The output is then compiled into a submission-ready format for regulatory agencies. Are you familiar with CDISC standards? How do you ensure compliance with CDISC in your work? Yes, I am familiar with CDISC, Clinical Data Interchange Standards Consortium, standards. To ensure compliance with CDISC in my work, I adhere to the CDISC data standards, which provide guidelines for organizing, standardizing, and exchanging clinical trial data. I follow CDISC SDTM and ADAM standards for dataset structure and variable naming conventions. I also ensure that the data transformations and analysis adhere to CDISC guidelines, allowing for better interoperability and regulatory compliance. You receive a dataset containing adverse events from a clinical trial. How would you use SAS to identify and summarize the most common adverse events? Sample answer. To identify and summarize the most common adverse events, I would first use SAS proc-freq to generate a frequency table of the adverse events variable. Then, I would sort the table in descending order based on the frequency count to identify the most common events. Additionally, I could use proc means to calculate summary statistics like mean, median, and standard deviation for continuous variables related to the adverse events. You are tasked with merging two datasets, one containing patient demographics and another containing laboratory results. How would you perform this merge using SAS? Sample answer. To merge the two datasets containing patient demographics and laboratory results, I would use the SAS merge statement. I would ensure that both datasets have a common variable, example, patient ID, that can be used as a matching key. By specifying this key variable in the merge statement, SAS will merge the datasets based on matching values. I would also select the appropriate type of merge, such as inner, left, right, or full, based on the desired outcome. You need to create a derived variable that calculates the age of each patient at the time of enrollment in a clinical trial. How would you accomplish this using SAS? Sample answer. To calculate the age of each patient at the time of enrollment, I would use the SAS INTCK function. First, I would subtract the patient's birth date from the enrollment date to obtain the number of days between the two dates. Then, I would divide the result by 365.25 to account for leap years to obtain the age in years. I would store this derived variable in the dataset for further analysis. You have a dataset with missing values in certain variables. How would you handle these missing values using SAS? Sample answer. To handle missing values in SAS, I would first identify the missing values using the missing function or the ISMissing operator. Then, depending on the analysis requirements, I would choose an appropriate method to handle the missing values. Options include deleting the observations with missing values, imputing the missing values with mean or median values, or using advanced techniques like multiple imputation or maximum likelihood estimation. You are required to create a safety dataset that includes only serious adverse events from a larger dataset. How would you filter and create this new dataset using SAS? Sample answer. To create a safety dataset that includes only serious adverse events, I would use a SAS where statement. I would specify the condition for serious adverse events in the where statement, such as ASAV is equal to serious. This would filter the original dataset and create a new dataset that contains only the serious adverse events. I could also specify additional conditions or combine multiple criteria using logical operators and/or in the where statement. You need to generate a table listing the demographic characteristics of male and female patients separately. How would you achieve this using SAS? To generate a table listing the demographic characteristics of male and female patients separately, I would use the SAS proc tabulate or proc means procedures. I would specify the demographic variables of interest as the analysis variables and the gender variable as a classification variable. This would produce separate tables or summary statistics for male and female patients, providing a breakdown of demographic characteristics by gender. You are given a dataset with duplicate observations. How would you identify and remove these duplicate observations using SAS? Sample answer. To identify and remove duplicate observations in SAS, I would use the SAS sort procedure followed by the data step. First, I would sort the dataset by all the variables that define a unique observation. Then, I would use the by statement in the data step and the first .variable and last .variable flags to identify and remove the duplicate observations. By outputting only the first occurrence of each unique observation, I would obtain a dataset without duplicates. You need to generate a report with aggregated results from multiple datasets. How would you combine and summarize these datasets using SAS? To combine and summarize multiple datasets in SAS, I would use the SAS data step with set statements. I would specify the datasets to be combined using set statements and ensure they have the same variables and variable types. Then, I would use appropriate statements like by, if, and retain to control the merging and aggregation process. By applying functions like sum, mean, or count, I could summarize the data across datasets to generate the desired report. You are working on a clinical trial dataset that includes data from multiple study sites. How would you create separate datasets for each study site using SAS? Sample answer. To create separate datasets for each study site in SAS, I would use the SAS data step with a by statement. I would sort the dataset by the study site variable and specify the by statement in the data step. Then, I would use the first .site and last .site flags to identify the beginning and end of each site's data. By using conditional logic and output statements, I would create separate datasets for each study site. You are asked to generate a graph showing the change in vital signs over time for different treatment groups. How would you create this graph using SAS? Sample answer. To create a graph showing the change in vital signs over time for different treatment groups, I would use the SAS sgplot or gchart procedures. I would specify the time variable on the x-axis and the vital sign variable on the y-axis. To differentiate the treatment groups, I could use color, symbol, or line styles. By using appropriate options and statements in the procedures, I would customize the graph to display the desired information and visualizations.